This is the YouTube demonstration for Introduction to Data Mining. This is the group Ida Lee, Wei Han, and Kevin Ma. And what we're going to be looking at is the data set right now, which we analyzed. We chose to analyze the Broad Institute Brain Cancer Gene Expression Level 2. So what we're going to do is we're going to take a quick look at the raw data set right now. So the Level 2 data set, oops, wrong editor. If we open up the level 2 raw data, you can see it's 1, 2, 3, 4, 5, 6, 7, 8, 10, 25 dimensions of data. And what we're going to do is we're going to do a dimensional reduction to reduce the amount of the dimensions which have little correlation with the rest of the data. This will allow us to actually visually display the data and see any validation of correlation amongst these data sets. So we're going to take this huge amount of data and we're going to make it look like this. This is an example of one of our LLE dimensional reduction outputs. And what we're going to do is we're going to show you first how we were able to obtain this data set.
So now we will do the dimensional reduction using our custom dimensional reduction component, which uses MATLAB. We use an API called MATLAB Control to manipulate MATLAB through Java. And so we will click this button and it will bring up MATLAB, which takes a few moments. So now MATLAB is initialized and we can select the data that we want to load into memory to perform the dimensional reduction. We can do either PCA, which is a linear reduction, or LLE, which is nonlinear. First we'll do PCA, then we'll do LLE later. We can also input the target dimensionality. So in this case we'll put 2 to reduce this data set down to 2 dimensions. So the PCA reduction finished very quickly, and it saved the output, which is basically 2 columns of data, into a text file to that same directory. And then we can then perform an LLE reduction using that same data set. LLE takes one parameter k, which is the number of nearest neighbors to use in the algorithm, and then we can keep 2 as the target dimensionality. And this algorithm takes a little longer to run than the PCA reduction. So now the reduction is complete and it's written out the 2 dimensions into a text file in that same directory. And so now we can load those data sets into the visualization component to interpret the data.
So this is going to be a demo of the visualization aspects of our data mining project. For the first thing we did is we actually visualized the data using Weka. We loaded up the actually raw data from the Baylor Institute, as well as the UNC, and compared the two, and just try to figure out which one we thought, what data set we wanted to use. So in the end, we chose the Baylor Institute data, and we're going to actually just visualize the level 1 raw data from both, just to give you guys, a peek into what we're doing when we're with our process. So using Weka, we can actually display a histogram of all 25 dimensions of data which we had with the Baylor Institute brain cancer gene expression. So just to shows you, the frequency of points which we had in each of these dimensions. This graph doesn't really show anything really interesting except the highest amount of a particular numerical value. For most of these ranges are the same, like 3 to about 14. This mostly represents the patients, I believe, and these data points represented different values of genes. I believe that was correct. And so next we also visualized the level 1 raw data using PCA dimensional reduction on the UNC, as well as the UNC level 1 reduction using LLE. So this is the PCA visualization. This gives a visualization somewhat like a comet, I would call it. There's an extremely dense packing of data points right here. Using JFree we are actually able to highlight over regions and actually zoom in. And if you mouse over to any of these, you can actually see the exact number of the points. And if we can actually zoom back out as well. So as you can see, the range of this data is ginormous. It's 1.8 million positive on the X side. We could highlight over this point and actually see exactly what value it is. And all the way down to about negative 15,000 on the domain. And on the range side, it goes from about negative 1 million to 500,000. And we're going to compare this to the dimensional reduction using LLE. So this is the LLE visualization. As you can see, the range and the domain have been significantly reduced. That's just how the dimensional reduction produces the output of that. And we can see this produces a sort of mushroom type shape. The LLE typically produced a tighter clustering of data, as opposed to where the PCA produces more of like a polygonical structure, as you'll see with our Baylor Institute data. So we can actually zoom in on these points and see, exactly where the clustering is occurring using this visualization. So we can, zoom down and we'd see this highly dense cluster right there. It's very user-friendly, and so there's about 22,000 points total, and as you can see, this cluster right here contains about 50% of that data point, of those data points. So we're actually going to zoom back out, and we are going to take a look at the Baylor Institute. I did PCA first, so we're going to take a look at PCA first for the level 2 data from Baylor Institute. So as you can see, the PCA data is very cluster-minded. The domain and range are significantly smaller than the LLE data set. If we zoom in, again, we can actually see where the clustering is occurring. About right here is where the majority of these points are that are produced from our dimensional reduction. And of course, again, we can highlight over these points and see exactly the size. And then we're going to take a look at the LLE. As you see again, it produces a very this actually produces a very linear this linear up to here display of the data points as well as here, and then it has this, I would describe this as some kind of seashell pattern. The majority of the points are actually centered right here. So you can zoom in actually on the cluster and see where these points are all gathering together. And we're going to show you a normalized version of the level 2 Baylor Institute data. So now we're going to take a look at normalized values for the level 2 Baylor Institute PCA and LLE. So we run a normalized function through this, and it scales up to the LLE data. And as you can see right here, the centers of these the center of these two data sets are fairly reasonably close to each other. It just shows that the clustering of points is approximately about here-ish. Could be, I guess visually we could see probably a better association if perhaps if we were to move these outlier, maybe some of these outlier points right here, or even some of these. So this is the normalized visualization of the level 2 data together, and we actually have a normalized version of the UNC and the Baylor Institute data. Let me show you that. So if we show the normalized version of all data points together, the level 1s and level 2s, we get a graph that looks like this. So this is on the scaling of, this was the I believe this was the LLE. Yeah, this was the LLE data. So it's the same scaling as the LLE, except everything is normalized. You see the same clustering is about here. Unfortunately, I wasn't able to implement a click method, so we can click on one of these classes and see that overlaid on top of the rest of them. It goes in order of 1, 0 first. So that's something in the future work which we could implement. So that is it for the visualization, and thank you very much.